Within the human body, the majority of cells go through replication at some point. The majority of the time the cell is alive, it's not replicating. It's just functioning to keep you alive. So what makes a cell want to replicate in the first place? Signals from something else. You know that Starbucks drink you always be wanting? They're never going to make it until you put in that order online. This signal is going to send the cell from G0, or that just functioning state, to G1, where it's going to start to grow. There are two different phases of growth. This is the first phase of growth, so G1. And this transition from G0 to G1 begins interphase of mitosis of the cell. G1 is the longest phase of the cell cycle. The cell has to grow. As soon as the cell receives a signal like, hey, you need to replicate, it's like, replicate where? We ain't got space for this. So the G1 phase is creating space for replication to occur, for that cell to now be able to have two identical copies of everything inside of it and not break. So the first thing the cell has to do is gather all of the materials to be able to do DNA replication. That requires making protein. At the end of G1, there is a checkpoint to make sure that everything is okay before the cell keeps progressing. We don't want to waste a lot of energy unnecessarily. So the end of G1 is both checking to make sure the DNA doesn't have any mutations. We're not about to replicate a mutation. We're going to send you back and fix the mutation before we move on. And two, making sure all of the proteins that will be involved in DNA synthesis, as well as any other transcription factors that help transcription occur. When we do this DNA replication, we're also going to be making proteins to be able to hold all of this DNA, and we need to make sure we have all of the materials ready to do all of this stuff at the same time. So now the cell has gone through the G1 checkpoint, everything's good. It's going to enter the S phase. S standing for synthesis. Synthesis of what? DNA. It's also synthesis of protein, because remember, the G1 checkpoint was also checking for transcription factors, which means proteins are going to be made during synthesis as well. The G1 checkpoint already looked for mutations. There's no need to have another checkpoint after the S. It can move on straight to the G2. In G2, the cytoskeleton, or the framework that keeps the cell from collapsing on itself, is going to be dismantled. It also gives the cell time to build up energy. It's about to go through another really exhaustive process, and it just used a lot of energy to get through G1 and S. So the G2 gives the cell time to regenerate the energy necessary for the upcoming mitosis to occur. G2 phase ends with another checkpoint, specifically checking for transcription factors, because there's a lot of proteins that are going to have to be made throughout the entire cell cycle. And if those proteins aren't made, the cell is not going to be able to divide correctly. The completion of the G2 checkpoint ends interphase, and the cell now enters the prophase of mitosis. Do you see what I did there? Now that the DNA has been replicated, it has to actually move somewhere. Prophase, or the first phase of mitosis, is where the cell begins dismantling all of the stuff that used to hold the DNA, aka the nucleus. All of the DNA that was synthesized during the S phase is unwound, really long strands of DNA. In order to make sure that that is organized to be able to go into individual cells, it has to be really, really tightly wound. Imagine having headphone wires. You need to wrap them to store them, as opposed to just crumbling up and putting them in your pocket. How much more work you have to go through to detangle them. So prophase consists both of condensing of the DNA and dissolving of the nuclear envelope or nuclear membrane. As the cell moves from prophase into metaphase, so prometaphase, there's going to be assembly of a kinetochore, which is a whole bunch of proteins together that are attached to a chromosome for the spindle fiber to come and anchor later. The cell keeps moving from prometaphase into metaphase, all of the DNA lined up in the middle of the cell. Now that this kinetochore has assembled around the chromosome, a spindle fiber can actually come and anchor into the kinetochore. This is going to provide the space for the chromosomes to actually start to get pulled away from each other. Another checkpoint occurs after metaphase to check for centromeric tension, or checking to make sure that that spindle fiber has anchored really tightly into the kinetochore and in the right place, and there's only one attached to each chromosome. If this goes wrong, you end up with new cells that have something known as an aneuploidy, or an unequal amount of DNA in the cell. Now we pass the metaphase checkpoint, we can actually pull things apart, separating the two chromatids from each other. A chromatid is one copy of a chromosome. You can have two copies of the same chromosome. One of those copies is known as a chromatid. Telophase returns what the locusts of prophase have took. You have this nuclear envelope beginning to reemerge. All of that DNA doesn't need to be tightly wound up anymore. It's able to start to unwind, and you're also getting a reassembly of the cytoskeleton. One part of the cytoskeleton is known as actin. Yep, actin from muscle. Actin filaments are going to form something known as a cleavage furrow in the center of the 1-2 cells. And because that actin, like from muscle, it's going to make a contraction. It makes a contractile ring that makes the cell break into two individual cells. Now, where once was one, there is two. Now that we understand how the cell cycle works, back to the patient's question. Is this cancer? Cancer happens when one of the cell cycle checkpoints goes awry or there's a mutation in one of the proteins of the checkpoint. So a mutation in either cyclin or CDK or a cyclin CDK complex or the retinoblastoma protein. Retinoblastoma is normally bound to DNA to prevent transcription. In order to allow transcription to occur, you got to remove that RB so you can get access to the DNA. And what removes the RB? Phosphorylation, meaning adding a phosphate group. Who's going to add the phosphate group? The cyclin CDK complex. So if there is a problem with the cyclin CDK complex or retinoblastoma, there's going to be an ability for cancer to occur because it can evade that first checkpoint. Similarly, in the G2 checkpoint, it's again checking for cyclin and CDK. This cyclin and CDK is bound to RB, but is also bound to other proteins. So if you have a mutation in cyclin, CDK, RB, or other proteins, you're again going to have the ability to cause cancer by evading the G2 checkpoint. You can also have something called a gain of function mutation, where something that wasn't supposed to function at all starts to now function. There are proteins that regulate cyclin and CDK complexes forming in the first place. One of those proteins is called P53. So if you have P53 that is mutated and it's causing too many cyclin or CDK complexes to form, that can also lead to cancer. But cancer is a mutation of some type of protein that has an impact on the cell cycle regulation. If this mutation does not have an impact of the cell cycle, it's not going to cause cancer specifically.